Peking University. Let's give her a warm welcome.
First, I'd like to thank Martin and also Alex Evelyn for the invitation, and also thank you for all the previous invitation that when I couldn't make it. So here, I'm going to give my second talk at this meeting, Measuring Aging with AI. Okay. For this audience, I don't need to argue that aging is quantifiable by aging clocks, actually largely attributed to Steve's work popularizing aging clocks. So aging clocks are essentially regression models that can measure regress to chronological age. And when you look at any individual, you can use the predicted age minus the actual age, the difference, to calculate a certain person's aging rate. Earlier, we have been using 3D facial images to build aging clocks. And we used, can we see this? Yeah. We used linear models as well as AI models using CNN framework. Compared to linear models, of course, AI models is much more accurate with an error of only 2.8 years versus the linear model 4.5 years on average. And we also trained CNN models to learn the perceived age given by people. With all of this aging rate, we can then couple them with the analysis of blood transcriptome. We can use the so-called causal inference analysis to ask what transcript in the blood transcriptome potentially mediates lifestyle impact on accelerated or decelerated aging on the face. For example, here we infer cigarette smoking, drunkenness, preserved food can all accelerate facial aging through enhancing inflammatory cytokines in the blood. On the other hand, there is several good lifestyles. In fact, it includes coffee drinking that can decrease facial aging rate through some other mechanisms. And what I just showed you are this on the forefront, on the foreground are the coding genes. But in the background, these are all the non-coding genes that we inferred to be mediators for accelerated or decelerated aging on the face. It looks many more than coding genes. That's why we decided to do a more comprehensive analysis on aging-related human link RNAs. What we found was that aging upregulated link RNAs often form positive feedbacks with NF-kappa B signaling. On the other hand, we also identified a very long non-coding RNA called CNQ1OT1 that decrease in early senescence. And this decrease leads to de-repression of transposon elements and cellular senescence. How does it do this? We found out through genetic and also computational analysis, it actually can sequence-specifically recognize transposon elements using the Hugelstein base pairing principle, forming the double-strand DNA and RNA triplexes. It can also simultaneously recruit the DNA methylation machinery, thereby it represses the transposon elements. Not only aging is written on the face, also some diseases are written on the face. For example, here we collaborated with ICU doctors, collected 200 stroke patients' 3D facial images, and together with 3 times more age and sex matched controls, we can then generate this stroke predictor, again using CNN model. It's very accurate with an AUC of 0.91. Even on independent cohort, it has an AUC of 0.82. And we also found that this predictor, the predictor given calculated stroke probability is correlated with age and also aging rate. What it learned on this phase is actually only very specific. It's only in the blue channel. In the 3D image, we actually use 4 channels, the RGB and also the depth. Only in the blue channel, we found there's a very strong signal. The central ballooning or central cyanosis is associated with stroke and stroke probability, stroke risk. In fact, even before stroke occurs, we can identify the mini strokes or the TIAs, transient ischemic attacks. Later, we have also been collecting the thermofacial images for over 2,800 people. Based on this, we developed an algorithm to normalize and compare different faces. Essentially, we first identify the landmarks on the thermal image, and based on that, we segment the image into around 900 triangles. And then, using this pattern, we can look at what are the changes over the course of aging. What we found is that the most significant feature associated with age is the temperature drop in the nose area and also the temperature increase around the eyes, which are illustrated here more clearly for both female and male. And also, we noticed that with age, the temperature variations on the face is increased, indicated by the standard deviation in each of this triangle. Of course, next we want to know what are the molecular events associated with accelerated thermofacial aging or decelerated facial aging, thermofacial aging. To do that, again, we did PBMC RNA-seq for 50 individuals, and then we coupled with a co-analysis with the aging rate on the thermal phase. We found that accelerated thermal aging are associated with herpes virus infection, DNA damage repair pathway. All of those are highly energy consuming. And on the face, they are associated with the increased temperature in the eye area. And consistent with thermal being related to energy and metabolism, we found that all these metabolic diseases have accelerated thermofacial aging, significantly accelerated thermofacial aging. And they also share this common increase of the temperature around the eyes, but also they have a unique feature that is the temperature increase on the side of the cheeks. Using this pattern, we can then classify hypertension, fatty liver, and some other metabolic diseases with fairly high accuracy with AUC over 0.8. On the other hand, decelerated thermofacial aging we found are associated with adipocyte lipolysis, proton ATPase. Both of them are heat generating processes. And consistently, we also see that the signature on the face is increased temperature at the nose. Unfortunately, some of the lifestyles we found are significantly associated with decreased thermofacial aging. One of them is sleep time. Sufficient sleep not only decreases the temperature around the eye and increase the temperature at the nose, but also more dramatically, it decreased the standard deviation over the whole face. And lastly, we also did a small intervention cohort with 23 females jumping rope. They are 18 to 49 years old. After 2 weeks, what we found was quite amazing. They have a thermal phase aging clock age decreased by, on average, 5 years. So after I look at that data, I decide to include rope jumping in my morning routine. In addition to using facial images, we also built single-cell RNA-seq clocks for the blood transcriptome. For this one, we collaborated with Dongfang Hospital doctors. We collected blood samples from 28 individuals and built this cell type composition clocks. The cell types contributing to these clocks are mostly the CD8 cytotoxic CD8 cells and monocytes that increase with age, and also many naive T cell, B cells decrease with age. Applying this clock to independent cohort samples, such as COVID-19 single-cell RNA-seq data, we found that even the mild COVID-19 can increase this clock age by 7 years, and the severe COVID-19 increased further by 10 years. On the other hand, for supercentenarians, those who live more than 110, we predicted them to be, on average, only 90 years old. And how did they do that? We found that in almost all cell types, they have very low cytokine, inflammatory cytokine level. And on the contrary, they have very high level of ribosome protein expression, even higher than many of the young people. In order to transfer learning to facilitate transfer learning of our single-cell clock to any other independent single-cell RNA-seq data from the blood, we also developed this transformer model to unbiased, without any batch effect, to directly transfer the cell type annotation. And also, at the single-cell level, we developed a program called Senescence, which is a program for identifying and quantifying single-cell senescence. We all know that senescence is very heterogeneous. So we collected a very large sample size, basically all the ones that are available, using SA beta-gal as the marker to identify senescent cells, and also these are coupled with the bulk RNA-seq. Altogether, we collected 600 samples from 52 studies, enclosing 30 cell types. Considering the heterogeneity of cellular senescence, we developed this program by first generating an SVM classifier for senescence versus non-senescence. I should say that in this case, the non-senescence including both proliferating and quiescent cells. And then if we then apply this SVM model to a second cell type, if the second cell type can be also well predicted, that is with an AUC over 0.9, we then classify this second cell type into the same seed type as the first one. The seed type is senescence ID type. By doing that iteratively until we exhaust all the 30 cell types, we found that 6 seed types can well classify all of the cell types. So what are the seed types? We found the genes that can distinguish the seed types actually rely on before they become senescent. So even before they are senescent, we can see this differentially expressed genes across different seed types, cell types of different seed types. And surprisingly, they are not related to any intervention methods, but they are related to cell type lineages. For example, the seed2 is skin-related lineage, and the seed3 is bone lineage, etc. So this then allows us to build another classifier. For any given data comes in, we first look at this data to see which seed it belongs to. We actually build another SVM model using these DGs. So first, we determine which seed it belongs to, and then based on that seed, we calculate a senescence score, seed-specific senescence score. Consistent with this lineage association, we also see that the senescence basal level, that is without being induced to senescence, the cell types senescence baseline are negatively correlated with stemness. That indicate for stem cells, they are much further away to senescence than differentiated cells, or that senescence is actually a continuation of the developmental or differentiation program. In fact, even though our SynSeed is trained on bulk RNA-seq data, with only one additional step, that is to impute the missing values in single-cell data, we can directly apply SynSeed onto single-cell data with very high accuracy, that is, again, over AUC over 0.9. With that, we can then look at the senescence trajectory at the single-cell level. First, we look at, for example, one cell type. This is the lung epithelial cell, 82 epithelial cell. During aging or chronic lung diseases or COVID, we can see always there's a dramatic increase in the proportion, the number, and also the level of senescence according to our SynSeed-2 score. We can also observe this clear trajectory of single-cell senescence. And during this trajectory, the upregulated genes and pathways are always the same, no matter we're looking at the aging or diseases, but the downregulated are very different. Also, the upregulated cell-cell interactions are mostly the same. However, if we look at different SynSeed types, cell types from different SynSeed types, we can also see this increase in just always during aging. We can always see there's an increase in the senescence level, and also there's clear trajectory. But the upregulated and downregulated pathways and genes are not the same for all these different cells, even though they're all during aging. And also the cell-cell interactions are not the same. Lastly, we also applied the SynSeed onto perturb-seq data to identify senescence modulators. Using a genome-wide perturb-seq dataset, we found that the senescence promoting perturbation targets largely aggregate into 8 modules. And these 8 modules correspond to all these essential cellular machineries, like all of this written here. But not all of them are equal. We found that this DNA replication, chromosome segregation module is actually downstream of all the other modules, or mediating the effect of all the other modules to impact senescence. Okay, with that, I'd like to stop here by thanking my students and postdoc who did the work, and also in collaboration with so many doctors in several hospitals. And lastly, we are recruiting international students and postdocs. If you are interested, contact me. And also our Center for Quantitative Biology at Peking University is recruiting new PIs. Thank you very much.
Thank you very much for a very interesting talk. Do we have questions?
Very, very interesting talk. You spoke a lot about the senescence in the cells, and you also said that you measure some aging in the cells. Is that correct? Did you do a clock for the cells?
For the senescence program, it's not a clock. It's actually, so what we did is the SVM model, so it's a class of, it's probability. Then we actually then use the regression, the logistic regression, to normalize the SVM probability so that all this probability is between 0 and 1. And this, actually our reviewers also asked us, why did you just do yes or no? Why do you do this scaling? In fact, the scaling means something. You can see during this trajectory, there's gradual increase in the senescence score.
So you would assume that the senescence score would also accelerate aging in the cells?
Whether they accelerate aging, we always observe senescence score increase with aging, but we don't know whether they are causal, which one is causing the other. Right.
Do we have more questions?
Really interested to hear about the jump rope results. Curious about two things. One, what was the duration of the rope jumping?
Oh.
And secondly, you said there was a 5-year decrease in the aging clock, but that was obviously an aggregate. Was that where was it for the 49-year-old compared to the 18-year-old?
We actually didn't observe there's any age bias in this. And for the duration, in fact, we require everybody to jump at least 800 jumps every day within, I think it's within 1 hour. It's pretty intense.
There's one question.
I want to follow up on that topic of the thermofacial clock. So all of the age-related trials obviously take forever. So can we use this technique as an early biomarker, whether somebody will be a responder or a non-responder in a metabolic trial?
Yes, we are actually using that in a few cohorts to detect some interventions. Yeah, but I cannot tell you what intervention we're doing. It's anti-aging intervention.
Can you comment on whether that can be a useful biomarker or not?
Yes.
For efficacy?
Well, according to me, I'd love to have this. I always have this 3D facial aging and thermal facial aging clock in all of our cohort because it's very easy to collect and it's very robust, very cheap. Our clinician collaborators, they really welcome it. You don't have to do anything extra. Yeah, so, but the thing is whether we can push it to acceptance by other people. We'll see. Now we do have a commercial application for it as well.
Okay, thank you.